Hi friends, we have a very short period of time for the semester examinations. Moreover, the immunology syllabus is vast and having very difficult concepts. So that's why I'm here, especially for those candidates who they are unable to study at the homes during these lockdown days due to books. So I am here to keep on uploading the concepts which are difficult to study. Okay, don't worry. So let's get moving to the topics. The MSc class 1, antigen, processing and presentation. Okay, in this lesson we're going to discuss about in detail each and every concept. First, what are MSCs? What are MSc class 1? And second, what they are also called as. And third one, the gene sequence of MHC, the structure of MHC, and final, the processing, the antigen processing. So let's start with what are MHCs. MHCs are nothing but these are cell surface proteins, okay, which carries an antigen to the cytotoxic T cells like CD8 positive cells. And what they are also called as, they are also called as major histocompatibility complexes, major histocompatibility complexes. And also called as HLA, human leukocytic antigens, human leukocytic antigens. And why the name is given as human leukocytic antigens? Due to, we all know that in an immune system or in an immune complex, the white blood cells plays a key role. They not only communicate with healthy and companion cells and also distinguish between infected cell and healthy cells. So what exactly allow these leukocytes to doing like this? So the reason is MHC molecules allow them to do so like this. And the gene sequence of the MHC. This is the gene sequence of the MHC. This is the gene sequence of the MHC. Okay. This is known as DP, this is DQ, this is GR. The scientists are named like this, so don't ask me about these gene locuses. And this is B on, this is B locus, this is C locus, and this is A locus. And this is MHC class 1 locus, which is present on all nucleated cells, expressed on all nucleated cells. Okay, this is class 2, MHC class 2, only express on APC cells, antigen presenting cells. And this is class 3 MHCs, class 3 MHC molecules. Okay, MHC, these are codes for cell surface molecules. Cell surface molecules are proteins, and these are also codes for also cell surface proteins. And the class 3 MHCs are codes for secretory proteins. Secretory proteins like interferons, complementary proteins, and cytokinins like others, and all such. Okay, and where are they present? In humans, these are present on 6th chromosome. Let's assume This is the long arm, this is the P arm, this is the Q arm, this is the paternal, this is the maternal. On the maternal chromosome, the MHC molecules are present, are situated. In mice, present on 17th chromosome. Okay, and the structure of MHC molecule. Now the structure of MHC molecule, just like this is the MHC molecule. This is the MHC molecule. It contains alpha 1 and alpha 2, beta 1 and beta 2 polypeptide units. Okay, alpha 1, alpha. This is the variable region of MHC molecule. Okay, it carries specific amino peptide fragments or amino acid ends which already affected or which already enters. Okay, it carries all those antigenic substances to the particular cytotoxic T cells for further clearance. Okay, now the antigen processing. Now we can see how antigen is processed by MHC molecules and together other cell components what are participated in and how they are helped to MHC molecule and what are proteins involved in it. We're gonna see here. So this is a cell. Let us draw. Example, this is a cell. This is cell. This is external fluid membrane. And internal fluid membrane. Membrane. Okay. This is the fluid. Okay, I completely draw. Let me draw completely. This is the fluid which is present in between the cell membranes. Now, let me draw some important cell organelles which are involved in and this is the ear and this is the ear. This is the endoplasmic reticulum which essentially synthesis proteins in this lumen. Okay, with the help of ribosomes. We're going to talk about all in soon. Now some important and interesting cell organelle, that is proteasome. And now Golgi apparatus, Golgi bodies. Golgi body. Before for the presentation, let us take an example of a pathogen, just like a bacteria. In this case, this is a bacteria. This is a bacteria. This bacteria will invade. It invade into a cell. Okay. Invasion. That means it penetrates into the cell, and the bacteria is multiplied in this cell and takes over the cell machinery and replicate itself more and more in number, takes over the cell machinery and infect the cell. It replicates and synthesis its own proteins. The bacteria synthesis its own protein molecules. Endogenous proteins of backs. Okay, now these proteins luckily will go through the proteasome, and the protein molecules which are entered into the proteasome, this is an interesting structure. This is in proteolytic inactivity, which digests endogenous proteins into a small amino acid molecules or peptide fragments. Now these endogenous proteins are broken down into 50 or 10 amino acid fragments. Okay, now these proteins have to be entered the lumen of endoplasmic reticulum. For the entrance, here a protein that helps to these amino acids into the lumen of endoplasmic reticulum. That protein is known as TAP protein. TAP protein. That means TAP means transporter associated with antigen processing. Okay, antigen processing. This TAP transport is coming from the gene locus of MHC molecules of the DQ arm, DP arm, from the DP arm. And this is the DQ, which contains 5 locuses, and DP is only one locus in a gene. This is DQ. Okay, the DP arm synthesis TAP protein. So this TAP protein will give us small clearance. Now these amino acid fragments are enters into lumen of endoplasmic reticulum. The ribosomes are get activated and synthesis a protein called MHC protein. This is MHC protein. This MHC protein has to be situated to the wall of endoplasmic reticulum. That can only possible by a specialized protein called calanexin. This is the protein called calanexin, and this is the MHC molecule. The MHC molecule which is synthesized by ribosome, it is incomplete due to it only contains 3 domains, particularly alpha 1, alpha 2 and beta 1. You might have got a question, where is the rest of beta 2? That will added in soon. Now a beta globulin the beta 2 molecule is formed and added to the incomplete and added to MHC in the presence of the protein called calreticulin. The protein calreticulin. Now this is calanexin. This is the calanexin, and this is the MHC molecule. Okay, this is calanexin protein. Now the protein is added to the side of MHC molecule, and this protein is slightly shifted the MHC molecule towards the tap 1. This is the portion tap 1, and this is tap 2. This is tap 1 and tap 2. Tap 2. These both and proteins are acting like a chaperones. Chaperone, that means that which protects MHC molecule and doesn't undergo degradation. Okay. Now this MHC molecule will shifted beneath, and it contains those proteins. Now the MHC molecules just like shifted towards to the tap 2 transport and migrate to the tap 1 site. Now the special protein called tapasin, the protein tapasin, is attached to the MHC molecule. And the function of calanexin is over, and it simultaneously degrades. It dissociates with MHC molecule. Only the calreticulin protein, which formed just like a protector which covers the variable region of MHC molecule, to do not attach with these amino acids of bags, because if those are attached, the protein will get attached with the amino acid fragments. Now the tap 1 transport shifted to the tap 1, the MHC molecule remain in the same position. The tapasin, and also when the tapasin molecule and calreticulin are just removed or dissociated, detached with MHCs, and these amino acid fragments are get situated. Now the MHC molecules is endosomed. Now the MHC molecule is endosomed by itself by endoplasmic reticulum. This is endosome. This is amino acid fragment. This is entered through Golgi bodies to the cell surface. Surface of the cell. When it comes out the surface of the cell, it has to be wait for the crosstalk interactions with other CD8 positive or cytotoxic T cells. Now let me draw. This is another cytotoxic CD8 positive cell. CD8 positive cells. This is alpha 1 and alpha 2, beta 1, beta 2. Now this cell, now this CD8 positive cell with its receptor, T cell receptors, it contains two receptors, alpha, beta. Now these two receptors are attached with this beta 1 and alpha 1, and they confirm that what being expressed this antigen to us, or it is any other cell or MHC molecules. Now after confirmation, the CD8 positive cytotoxic T cell, this is CD8 positive cytotoxic T cell, it brings up its MHCs into the action. MHC will being expressed this antigen to us. When confirmed, the MHC molecules of CD8 positive cells will get attached with these MHCs, and some little crosstalk is also occurred. Then the fragment is shifted to this cytotoxic CD8 positive receptors, and then it gives and it releases chemicals or cytokinins. This is infected cell, and this is killer cell. Okay, and this, now the CD8 positive cell, it triggers or it elicits and sends some interference or cytokinins, such types of chemicals which kill the infected cell. This is the process of antigen processing and presentation. Thank you for watching.